Have you ever wondered how scientists can peer inside a living brain and watch its thoughts unfold, or observe which regions activate during a task? It feels a bit like science fiction, yet it is a reality that has revolutionized our understanding of the human mind. The blood oxygen level dependent, or BOLD, signal is the cornerstone of functional magnetic resonance imaging, known as fMRI essentially. It is an indirect measure of brain activity, detecting changes in blood oxygenation and blood flow that are tightly linked to neuronal action. The core mechanism behind the BOLD signal is called neurovascular coupling. When a particular area of your brain becomes more active, its neurons demand more energy. This increased demand triggers a localized increase in blood flow to supply oxygen and glucose. Interestingly, the brain overcompensates, sending more oxygenated blood than the active neurons actually consume. This surplus of oxygenated blood changes the local magnetic properties of the tissue. Hemoglobin, the protein in red blood cells that carries oxygen, behaves differently depending on whether it is carrying oxygen or not. Oxygenated hemoglobin is diamagnetic, meaning it has very little interaction with a magnetic field. Deoxygenated hemoglobin, however, is paramagnetic, which means it slightly distorts the surrounding magnetic field. Functional magnetic resonance imaging scanners detect these subtle differences in magnetic properties, which are then used to create detailed maps of brain activity. So, how do we use this information? Functional magnetic resonance imaging measures these changes in the BOLD signal over time to infer which brain regions are more or less active during specific tasks or in different states. It allows researchers to pinpoint areas involved in memory, emotion, language, and countless other cognitive processes. An important nuance to understand is the hemodynamic response function. This describes the characteristic sluggish and delayed nature of the blood flow response to neural activity, typically peaking about 5 to 6 seconds after the neurons fire. Because the BOLD signal measures blood flow and oxygenation rather than direct electrical impulses, it is considered a surrogate or indirect measure of neural activity, providing a window into brain function at a macroscopic level. Understanding the blood oxygen level dependent signal is key to appreciating how we map the brain's functions. It allows scientists to explore the neural underpinnings of cognition, understand brain disorders, and even monitor the effects of interventions. This remarkable physiological phenomenon provides a powerful, non-invasive way to observe the brain in action, continuously expanding our knowledge of its incredible complexity.